Hello, welcome to Study Hub Pro. So, have you ever thought about whether an organism could get new genes without, being born with them? Well, it might sound like science fiction, but in the world of microbes, it happens all the time. Today, we're diving into one of the most fascinating tricks in all of biology, a process called transduction. It's how bacteria get brand new DNA, not from their parents, but from their neighbors. And the couriers for this genetic information? You're looking at them, viruses. Specifically, these things called bacteriophages, which are pretty much nature's own microscopic genetic delivery trucks. So here's the game plan for today. We're going to start with the basics of genetic exchange, then we'll meet our key players, the bacteriophages. After that, we'll get into their two very different life cycles, explore the two types of transduction that can happen, and finally, we'll see how this amazing natural process is actually used out in the real world. Let's jump in.
Okay, so to really get a handle on transduction, we first need to zoom out a bit and look at the bigger picture of how bacteria share their genetic blueprints. We usually think of genetics as something passed down from parent to child. We call that vertical gene transfer. But bacteria are the undisputed champions of something called horizontal gene transfer. This means they can share genetic info directly with each other right now in the same generation. It's a huge reason they can adapt so incredibly fast, sharing really important traits like antibiotic resistance.
And bacteria have three main ways to pull this off. First, there's transformation, where a bacterium just kind of slurps up some free-floating DNA from its environment. Then there's conjugation, which is like a direct handshake, where DNA is passed through physical contact. But the one we're focused on today is, in my opinion, the coolest of them all, transduction, where a virus acts as a sort of accidental middleman.
Now that we've got the background, let's zoom in and properly introduce these viral messengers that make the whole process of transduction possible.
I love this term, molecular hijacking. It just perfectly describes what viruses do. See, they're what we call obligate intracellular parasites. That's just a fancy way of saying they are completely helpless on their own and need a host cell to replicate. They carry just enough genetic material to break into a cell, hijack its machinery, and turn it into a non-stop virus-making factory.
Now, viruses can infect pretty much all forms of life. But for our story, we only care about the specialists, the ones that exclusively target bacteria. And these are called bacteriophages, or just phages for short. The name literally means bacteria eaters. Just look at the structure of this thing. It looks like something straight out of an old sci-fi movie, kind of like a lunar lander, right? That head part is a protein shell called a capsid, and it's basically a little safe protecting the viral DNA. The tail is this really sophisticated biological syringe. Its tail fibers lock onto a specific bacterium, and then the sheath contracts and injects the viral DNA right inside, leaving the empty shell behind. Pretty incredible.
So, to understand how these phages can accidentally transfer bacterial DNA, we first need to get a grip on their two main strategies for infection, their two different life cycles.
All right, first up is the lytic cycle. This is the aggressive, fast-acting strategy. And a quick heads up, this is a super common topic on exams, so you'll definitely want to remember this process. It all starts with the phage attaching and injecting its DNA. The viral genes immediately go into takeover mode, shutting down the host's own functions and chopping up its chromosome. The cell is then forced to just replicate viral DNA and build viral proteins. All these parts then self-assemble into hundreds of new phages until the cell is so packed that it just bursts. Or, to use the scientific term, it lyses, releasing a whole new viral army. But not all phages go for this immediate, all-out assault. Some of them, which we call temperate phages, can actually choose a much stealthier path.
And this is the lysogenic cycle. In this case, after injecting its DNA, the phage doesn't destroy the host. Instead, its DNA actually integrates right into the bacterial chromosome. This little piece of integrated viral DNA is now called a prophage. It's like a secret agent hiding inside the cell's main blueprint. The host bacterium, now called a lysogen, just goes about its business, living and reproducing, and every time it does, it copies that hidden prophage along with its own DNA. It's the ultimate stealth mode. That is, until some kind of stressor, like UV radiation, triggers the prophage to pop out and kickstart that destructive lytic cycle.
So, what's the bottom line here? The lytic cycle is like a blitzkrieg. It's fast, violent, and always deadly for the host cell. The lysogenic cycle, on the other hand, is a long-term infiltration. The virus just lays dormant inside the host's genome, waiting for the right time to strike. You absolutely have to understand these two different paths to understand transduction.
Okay, so now that we know the official playbooks for these phages, let's look at what happens when things go a little wrong. Because at its heart, transduction is really just a packaging error that happens during one of these cycles.
First, let's talk about generalized transduction. And just like the name suggests, this is an error that's tied to that chaotic, fast-paced lytic cycle.
So remember how in the lytic cycle, the phage chops up the host's chromosome into tiny pieces? Well, in the middle of that frantic assembly line of building hundreds of new viruses, a mistake can happen. Instead of packing the viral DNA into a new phage head, the machinery accidentally grabs a random fragment of the host bacterium's DNA. The phage that's built is defective, but it can still infect another cell. And when it does, it injects bacterial DNA instead of viral DNA. So no new infection starts, but it can deliver brand new genes to the recipient cell.
Now, the second type of error happens during the much more precise, but still not perfect, lysogenic cycle. And this one is called specialized transduction. This is a great visual of the exact moment the mistake happens. Remember, the prophage is integrated at a very specific spot on the host chromosome. When it gets the signal to leave and start the lytic cycle, it usually cuts itself out very cleanly. But sometimes the excision is just sloppy. It cuts itself out wrong, and in the process, it grabs a little piece of the bacterial DNA that was right next to it, while leaving some of its own DNA behind. This really shows you why the phage's integration site is so important. Okay, this slide right here, this is perfect for your notes. So let's really break it down. Generalized transduction, that happens during the lytic cycle, and it can transfer any random gene from the host. It's a total crapshoot. Specialized transduction, on the other hand, is an error from the lysogenic cycle, and it only transfers the specific genes that were physically next to the prophage. Because of this, the defective phage in generalized transduction carries only bacterial DNA. But in specialized, it carries a hybrid of both phage and bacterial DNA.
So, a process that starts as a simple viral mistake actually has enormous real-world implications. Let's wrap up by looking at how we've cleverly taken this natural process and used it for our own benefit. Believe it or not, scientists have turned this natural delivery mechanism into one of the most powerful tools in molecular biology. We use modified, harmless viruses as what we call vectors to deliver genes for gene therapy, to help us map out bacterial genomes, to genetically engineer plants and animals with new traits, and even to create specific mutations in bacteria just so we can study them. And this visual shows you exactly what that looks like in action. For something like gene therapy, scientists can package a healthy copy of a gene into a modified virus. This viral vector then does what it does best. It finds a target cell and injects that genetic material. The cell can then use this new functional gene to make the correct protein, which could potentially treat or even cure a genetic disease. It's amazing stuff. So what really started as a viral oops has become one of our most powerful tools for manipulating genes. Which leaves us with a final, pretty big thought to chew on. If viruses are nature's original masters of molecular hijacking, could they also be the ultimate key to correcting our own genetic errors? See you in the next video.